the study, essentially what we're trying to identify is common reproducible markers of the T-cell receptor repertoire in patients that we can use to potentially guide future treatment and also stratify patients who are going to do well or not well with immune checkpoint blockade. So we looked at CD8 T-cells from 91 separate patients and sequenced their T-cell receptors using very deep sequencing, on average identifying over 7,500 individual clones per patient. What we were able to do is, using certain algorithms, group those clones into those that potentially shared same antigen specificity, so called specificity groups. We took these specificity groups and took those groups that were found in more than 3 patients and matched to more than 3 T-cell receptors. And we looked at those which expanded in patients when they'd had checkpoint blockade and only expanded in patients with a good long-term clinical outcome. And that was defined as continued response to immune checkpoint blockade more than 6 months after starting treatment. Doing this approach, whilst we sequenced over 2.5 million clones in total, we whittled these clones down to about 3,500 specific T-cell receptor clones. And we could see that these clones, the proportion of the repertoire in the patient's peripheral blood that these clones occupied was much lower in patients who progressed prior to 6 months versus those who had an ongoing clinical response. And we could see that both pre-treatment and after treatment. So we further explored these clones, and we could see in a number of patients we had previously resected tumours. And we could see that these clones, whilst the tumours were resected over 6 months on average prior to starting checkpoint blockade, we could see that these clones were present in the tumours and in fact enriched in the tumours versus the blood. When we looked at clones that were both in the blood and in the tumour, they were much bigger in the tumour. And finally, from a subset of patients, we had single-cell sequencing, RNA sequencing. And so we could say, what do T-cells carrying these clones, what is the phenotype of those T-cells? And we found that in general, they tended to be central memory or effector memory CD8 T-cells. And when we looked at those effector memory and central memory CD8 T-cells that carried the T-cell receptor, they tended to have much more expression of cytotoxic genes, which was indicative that they were potentially more active. So finally, we took a further cohort of patients, again treated with for metastatic melanoma in Oxford with immune checkpoint blockade. So a further 57 patients, again purified the CD8 T-cells, sequenced the T-cell receptors. So this was a second replication cohort. And we could find that in patients who had an above average amount of their repertoire, so we'd split the cohort into 2, above and below average amount of repertoire occupied by these emergent respondent clones. And patients who had an above average amount of the repertoire occupied by these clones had an overall survival benefit and a progression-free survival benefit. And we finally we looked in further other data sets, so the RIAS data set from 2017, and we could replicate the same finding. Not presented in the talk, but we can see that these clones show HLA specificity, and then we look at the patients who have a long-term clinical response, i.e. many years, we can see these clones persist in their peripheral blood.